Alrighty. Well, hi guys. I'm Grant. I'm one of the grad students in Dr. Vipa's molecular biology class in Bowling Green. I was helping a couple people earlier try to explain activators, repressors, and Foxp3 because it's one of the more complicated things we're covering for this exam. And I figured I'd share the wealth and try to do it Khan Academy style, upload it to the canvas so you all can see sort of my oversimplified explanation of Foxp3 activators and repressors because they're all kind of inherently related. So as you see right here, this is my DNA. This is what DNA I would like or not like to transcribe. So you'll see I've got my P is my promoter, my GOI is my gene of interest. So let's just say this produces pro-inflammatory cytokines. Well, let's say this produces anything. We'll start simple. Say this is a gene of interest I either want to or don't want to sort of transcribe. And as you'll probably remember, the general mechanism of transcription, which I'll make in, let's do purple. I'll have my RNA polymerase, RNA pol. My RNA polymerase will land on the promoter and the gene of interest and spit out a pre-mRNA. Now, this is a little too simple, so we'll make it a little more complicated, right? So actually, RNA polymerase doesn't necessarily work alone. It can have friends, which I will call collectively the mediator proteins. Mediator. So mediator proteins, effectively, they help RNA polymerase transcribe things, right? Okay, well, we'll make it slightly more complicated. Yay. So, also helping transcribe things are transcription factors. So, a transcription factor, which I'll draw over here on the left, this is basically an activator, which will help transcription, or a repressor, which will slow down transcription. It's got two domains. We'll say domain 1, domain 2. Domain 1 on my transcription factor, domain 1 is the DNA binding domain. So, if you look over here in the middle, I've got a little sequence of DNA called an enhancer. Now, enhancer is just a strip of DNA that is complementary to the DNA binding domain of an activator protein. So here's 1, 2. This is an activator. Activator. Your enhancer is usually upstream of your gene of interest. So you can say you have to travel quite a long ways. You have to travel quite a long ways from your promoter and your gene of interest all the way wound up. Boom, here's my enhancer. I've made it a little simpler by bending the DNA on itself. So as you remember, even though we draw DNA as a 2D structure, it's really a 3D structure. And a lot of times it can be folded on itself. It can be manually folded on itself more to speed things up. But sort of regardless of that, this is closer to the real 3D structure of DNA. So this upstream enhancer is complementary to the DNA binding region of an activator protein, which is a transcription factor that speeds things up. So let's go back to domain 2. So domain 2 down here, this is a activator or repressor domain. So it is what it sounds like. So domain 1, this is what does the actual work. Well, sorry, I should say this is what binds to the DNA. And domain 2 does the actual work of signaling to activate the mediator protein in the RNA polymerase or to repress it through a couple different mechanisms. And so the alternative, you can have an activator or a repressor. Activator speeds it up, repressor doesn't. That all depends on your domain. So we have a repressor, for example. It would bind to a silencer. It's S for silencer. So here's my repressor. But we won't worry about that yet. 1, 2. Okay. Well, how does my activator activate? So let's say I want to speed up whatever this gene of interest is right here. I want to make this go faster. So I have my mediator attached. Things are going well, but it's not going fast enough. So there's a couple different ways an activator can activate. So I'd say the simplest one, say your activator will send out sort of intermolecular signals to the mediator and the RNA polymerase. They're just like, okay, hurry up, do this faster, right? That's pretty intuitive. It's very close to it, so the proximity will make it work faster. That's why it's wrapped around on the enhancer. But yeah, first mechanism, it will send signals, be like, okay, hurry up, do this faster. Another mechanism is it can recruit what's called a co-activator. So let's say, one mechanism is the signaling to RNA polymerase in your mediator proteins. Say another mechanism, you shoot another signal to a co-activator. So a co-activator is just another protein associated with the activator. So a co-activator. And we'll use Cat5, the enzyme, in this example. So a Cat5 is an acetyl transferase. Acetyl transferase. Again, ACE is an enzyme. This means it will add acetyl groups to something. In this case, the something is a histone. So you see our nice DNA here, but a more realistic portrait of DNA with it being wrapped around a histone. So what color is my DNA? It's orange. So I like to think about a histone. I'll write that down. Histone. It's like a ball your DNA is wrapped around, but for this, I think it's fun to think of as an airport. So imagine your histone is an airport, and your DNA are the runways in that airport, and your RNA polymerase is a plane that's trying to land on the runway at the airport. Well, let's say a histone by default, let's say the lights aren't on. No one's turned on the lights at the airport, so theoretically the airplane or the RNA polymerase can land on the runway of the DNA, but it's a bit difficult. So that's why you get a co-activator like Cat5 and acetyltransferase, which will basically go and they'll add acetyl groups to a histone. So acetyl groups in real life, they open up the chromatin to make it easier for RNA polymerase to get in. But in this scenario, I like thinking of it as turning on the lights at the airport. So all these little acetyl groups, just imagine these are the lights at the airport. Oh, boom. It's lit up super well. My RNA polymerase, I'll draw arrows. So let's say, Cat5 will turn on the lights. So now it's like, oh, perfect. I can land. I can transcribe things really well. Thank you, activator and co-activator Cat5. So that's all jolly and good. So those are sort of your two mechanisms of activators. So the first one, the simple one, it will just send intermolecular signals to your mediator and your RNA polymerase. Be like, okay, hurry up. I'm very close to you. I'm going to get you to speed up. And the other mechanism is it will get a co-activator like Cat5 to do histone modifications. So this is a type of epigenetics that we talked about. In this case, the histone modification, you're acetyling it, you're turning on the lights at the airport. It's easier for the plane to land on the runway. So let's clean up some of this. now we talked about our activator. Let's talk about repressors. So a repressor is another transcription factor, but this time it binds to a suppressor, and shockingly, it suppresses/represses transcription. So there's 4 different mechanisms of repressors. So I'll draw a couple more silencers over here. So the first mechanism is you can sort of overlap an activator. So let's say here's my enhancer, and I have a silencer right next to it. Get rid of this. So my silencer, okay, let's say I don't want to enhance. I don't want the activator to bind. I will have a repressor, again with those 2 domains, bind to my silencer. And then, oh no, my activator is confused. It can't land on the enhancer site. So it's the first mechanism, it's competitive binding. So it'll overlap that site. Okay, well, I can't land here. I guess I can't be activating. A second mechanism is, let's add a new, say it can bind to the mediator. So this is called direct repression. So let's say I have a particularly long repressor. My repressor will bind to the mediator, and this is just like the first mechanism of an activator. It will send intramolecular signals and be like, okay, stop it. Bad. I need you to stop transcription. So this is like the evil version of an activator. So that's pretty intuitive. You can also bind near the promoter. So let's say I shouldn't have made the promoter red. Let's clean this up over here. This is a special suppressor. I'll make this one orange. So no, maybe not orange. Taking that too. All right. Well, we'll make it red. Use your imaginations. So let's say I have another silencer right here. Say my repressor binds, and it can block the promoter. So actually, my mediator and my RNA polymerase, when this activator is bound, can't even bind. So they'd see that promoter and be like, oh, there's something blocking the road. Well, that sucks. I can't bind here. And they're not going to associate. And they're not going to do transcription. It's kind of unfortunate. But, job well done. All right. And mechanism number 4 sort of goes back to this histone modification. So let's say I have my repressor bound to the silencer. This can send 2 signals. So let's also say I have an activator here. So here, I'll move the suppressor to not confuse it with the site overlap. All right, here's my repressor. There's my activator. Say my activator is doing its thing, making Cat5 to turn on the lights at the airport. Well, the first thing my repressor can do is like, hey, Cat5, don't do that. And I like to think of it, let's say it's a dog whistle. All right, stick with my analogy here. Your repressor sends out a dog whistle that will just make your Cat5 co-activator go deaf. So Cat5 is like, I can't hear anything. I'm not doing my job. I'm not going to turn on these lights at the airport. I have bigger problems like my ears bleeding from this repressor dog whistle. At the same time, another mechanism of repressors, it will send another signal out and around to a co-repressor. In this case, we'll use HDAC. HDAC stands for histone, poorly written, deacetylase. So the HDAC is an enzyme, again signaled by the repressor binding, which will turn off the lights at the airport. So HDAC will go through, send a signal, and start slowly deacetylating my histones. Now again, deacetylating, let's say, imagine HDAC is a dog at the airport. And me, the repressor, I've trained my dog super well that I can use this. The same dog whistle will deafen the security guards, Cat 5, trying to turn on the lights. And it will have my HDAC dogs who are going to run on the runway and chew up all the lights. So boom, my RNA polymerase thinks he's about to land on the runway. That's my DNA. But actually, all the lights just got turned off by HDAC. So it's a deacetylase. So it's removing the acetyl groups, which you can think of as turning off the lights on the runway at the airport. So not only does it turn off the lights, it suppresses Cat 5, which is trying to turn the lights back on. So, if you don't suppress Cat 5, it would be silly because you turn off the lights, Cat 5 comes along, turns the lights back on, you're in a silly little battle. So it sort of has this mechanism to stop them from turning back on. And again, RNA polymerase can't land. It's not going to transcribe anything. So that's a pretty general good mechanism. Those are the four mechanisms that repressors can work and sort of the two mechanisms of activators. You're like, okay, well, what does this have to do with Foxp3? So let me clean up my whiteboard a little bit. Maybe I want to keep my 5' up there. 5'. It's a little bit messy, but I mean, you guys can figure it out. All right. Back here, and I'll draw out my Cat 5. We'll do it over here. Or sorry, not my Cat 5. Foxp3. So Foxp3 is I'll draw it sideways at the bottom here. That should be easy to remember. Foxp3 down here is a fork-head box protein 3. And it's got a couple different regions that are important to know. Let me grab me out my cheat sheet. So one region, it's pretty easy. This will just be your DNA binding region. So if you imagine it, as a big circle like the others, here I'll draw the circles over this. Domain 1, domain 2. So domain 1 is your DNA binding. Now, a unique thing about Foxp3, write that, Foxp3, is it's a transcription factor that can act as an activator and a repressor. So a way I like to imagine the DNA binding region here, this isn't necessarily accurate, but I think it's a helpful way to remember. Just imagine one side of it can bind an enhancer, where it will act as an activator. Imagine the other side will bind to a silencer, where it will act as a repressor. So it binds to an enhancer, it's an activator. It binds to the silencer, it's a repressor. It just depends on which side the DNA binding you land on. Again, not exactly true, but it's a helpful visual way to remember that this is a thing that happens. They can bind to either of them. And then you have a couple different domains. Actually, here, and I'll make this a 3 blob. That'll be even simpler. All right. Here's domain 2. Here's domain 3. All right, so domain 2 we'll just call the activator domain. So there you have 2 regions. Or I should say, sorry. Let's call 1, 2, and 3. We'll call those regions. We'll call these smaller ones inside domains. these are called functional domains. So functional domain 1 is your DNA binding thing. This is sort of in region 1. So your DNA binding region just has one domain. It's just your DNA binding domain. Pretty simple. Let's call this region 2 is your activator region. So we'll say the activator region carries 2 different activator domains. Then region 3 will be your repressor domain, where it carries one big repressor. So I'll clean up my bubbles. So just imagine you still have your DNA binding domain, but you have a DNA binding region, like a normal transcription factor, but you have both an activator and repressor domain. So it can work at both. Just imagine, depending on where it lands, or which side it lands on. All right. So we'll call our first activator domain here is a leucine finger. Now, you don't need to know exactly what the leucine finger does, but you just need to know, okay, it's just intracellular signal. We'll say this for the next one too. Next one is a zinc zipper. Oh, sorry. Flip those around. We have a leucine zipper and a zinc finger. So you think zinc zipper, it's a much nicer name. Oh well. I don't name these things. Very important Nobel Prize people name these things. So these both work largely, but they can either recruit enzymes like Cat5, which we talked about ad nauseum, to go and acetylate the histone, or they'll just send signals sort of to the mediator in the RNA polymerase. Be like, okay, well, hurry up, do your job, transcribe this gene. So in Cat5, usually the gene you're transcribing is sort of repressing a gene that makes a protein that represses the immune response in T cells. Okay, so as an activator, let's say it binds on my enhancer to my Foxp3. Send signals here, get going. You'll also send a signal to add acetyl groups and turn on the lights at the airport. Too easy. Well, let's say it binds to the other side. Binds to a silencer. Now it's going to act as a repressor. So I'll draw someone angry and red. So as a repressor, its main job is it's the histone modification mechanism of repressing. So we talked about, maybe it'll repress by interfering with the enhancer and activator. Maybe it'll block the site here. Maybe it'll reach down and you directly interact with the mediator in the RNA polymerase. In this case, Foxp3 as a repressor just recruits lots of co-repressors. So the big one to remember here, which I'll do in this purple, is HDAC. So remember, HDAC turns off the lights. It will also suppress Cat5. So it's the same suppressor mechanism we talked about. So really, they only, all right, this is the repressor domain. So really, the most important thing about Foxp3 is it's not very intimidating as long as you understand how activators and repressors work. So think Foxp3 does both. So it's got this leucine zipper, this zinc finger, that work as an activator domain. So let's say my DNA side lands on the enhancer. Okay, I'm going to activate. Let's say it's having an off day. It's like, actually, I want to repress. It'll land on the silencer and then act as a repressor through recruiting these things to modify histone. So recruit HDAC, block Cat5, and then your histone, hopefully at your histone airport, there's not very many acetyl groups. All the lights are off. No RNA polymerase can land. So you send a dream world of the repressor domain. None of this even happens. No mediators, no repressor. It's doing such a great job. that nothing gets transcribed. So really, the main function of Foxp3 is to modify T cells. Let's make T cells blue here. Say this is a T cell. T cell. Or I should say this is we'll call this a T regulatory cell. That's more accurate. So I'll call this a T reg. So T reg is like your immune system quarterback. Or let's say it's your immune system general. So T reg, like, okay, immune system, there's something going wrong. There's an antigen. I need you to destroy this thing. Or be like, okay, well, I need you to stop destroying this thing. But really, T reg itself isn't making these decisions. It's your body and Foxp3. So Foxp3 will just, if it's activating, it'll tell T reg, okay, I'm in the nucleus of T reg. I need you to start making, I don't know, let's say suppressor proteins. Suppressor proteins. So I need you to, I'm turning on the gene of interest for suppressor proteins. Say, okay, suppressor, I need you to go out and I need you to slow down my immune system. It's doing too much damage. Let's say it can also repress pro-inflammatory genes. So let's say you have a very angry, let's just say inflammatory gene. We'll say there's inflammatory genes on the Treg, just for simplicity's sake. Here's an inflammatory gene. Basically, this will signal, okay, no, don't turn that on. Keep that off. I don't want any inflammation. Things are fine. Slow down. Call off the troops. So if you think about this, that's why mutation of Foxp3, this can lead to autoimmune diseases. So really, the gist of Foxp3, whether it's an activator or a repressor, its whole job is to calm down your immune system. You don't want your Tregs, blowing everything up all the time. The war has to end at some point. So let's say my Foxp3 is broken. So it could be a SNP. Let's say I have a mutation in my DNA binding region, and I can't bind to enhancers or silencers at all. So really, there's no chance it lands. Well, now there's no one signaling, oh, make suppressor proteins or stop making inflammatory proteins. It's just like you're toast. If it can't bind, there's nothing you can do, and your Tregs are going to be just on a crusade throughout your body. They'll always be making your immune system active. You'll always have inflammation. You'll have cytokines all the time, and that can lead to these autoimmune diseases like IPEX. So let's see. IPEX syndrome. But yeah. So again, I said, when we're talking about, is it a silencer or an enhancer? I made it sound kind of arbitrary which side it lands on. It's a little more complicated than that. It depends on, okay, well, what is the cell like? Is the cell, are other signals telling me, okay, I need to make suppressor proteins? Am I getting other signals saying I need to stop inflammatory things? Sort of the conditions in the cell will make it more favorable to bind at an enhancer or bind at a repressor. And that way it keeps everything in check. Or sorry, not a repressor. Bind to a silencer. So again, a silencer is the DNA region. A repressor is the protein that slows down transcription. So I know this was a bit chaotic, but hopefully this can be helpful to some of you in understanding Foxp3 as well as activators and repressors. This is a pretty important concept for the exam. And yeah, I hope this can be helpful.